Okay, so we can move on to Professor Bhattacharya from the Karolinska Institute with his talk about biocompatibility of nanomedicine approaches for arthritis, focus on carbon nanotubes. Thank you, Kunal.
Thank you, Dr. Bottini. Good morning, everyone. So as Dr. Bottini highlighted, I'm going to speak about biocompatibility of nanomedicines, approaches for arthritis, focus on carbon nanotubes. So first of all, I would like to start by thanking the CLINAM Foundation and Dr. Bottini for giving me the opportunity to speak in this conference. Dr. Bottini had, in the previous presentation, had really given a very brief outlook on the different nanomaterials that are available for treating osteoarthritis, such as the liposomes, myoceles, and the carbon nanotubes, and how they can be enhanced and the drugs can be loaded, and targeting the chondrocytes in the articular cartilage. So I'm not going to go through all these things. So why carbon nanotubes? Carbon nanotubes are quite well known specifically in the field of nanosafety as inducers, which can act on the sensors or the innate immune cells to stimulate the production of mediators such as chemokines, cytokines, and complement factors that can lead to inflammation in the target tissues where they are exposed. However, these same cells also produce other type of enzymes which are known as peroxidase enzymes, like the macrophages and the neutrophils produce myeloperoxidases, and the eosinophil produces eosinophil peroxidases. These peroxidase enzymes, along with reactive oxygen species and reactive nitrogen species, which are also produced by these cells, can get attached to the carbon nanotubes when they are functionalized and degrade them into carbon dioxide and water as the final product. And it has also been studied by Swedova et al. That these byproducts that come out of the biodegradation of the carbon nanotubes are literally non-inflammatory into the cells. In 2014, we published a paper in nanoscale where we also demonstrated that we use several pegylated single-wall carbon nanotubes of different molecular weights and functionalities and used ex vivo humans neutrophils and saw that all these nanoparticles, carbon nanotubes, oxidized and the pegylated ones, were biodegraded by the neutrophils that is coming from the humans within 8 hours. So it was a very fast process of biodegrading the nanotubes. What we used for this analysis was a Raman spectroscopy, which is highly defined for looking at oxidized defect sites on the surface of carbon nanotubes. What we also found out very interestingly as a mechanism of action was that the neutrophils, when they degranulated, produced neutrophil elastase, and this was capable of cleaving the PEG chains from the surface of the nanotubes. And from these cleaved sites, the myeloperoxidase, when produced by the neutrophils, were capable of degrading the nanotubes. So we found a very good mechanism to see how the carbon nanotubes can be degraded by neutrophils. Following this, Dr. Bottini had already shown that they use pegylated carbon nanotubes, which were coated with 2 kilodalton PEG chains, to deliver antisense oligomers into the knee joints of diseased osteoarthritis mice and deliver the payload. So the question that came after this study was what happens to the carbon nanotube? Because we are talking a lot about nanomedicine, so we also have to look at the clearance mechanism for these from the body. So what we wanted to study after this was the trafficking of the pegylated single-wall carbon nanotubes in the knee joints of the mice. Are they biodegraded? And finally, if they are degraded, what is the mechanism behind this biodegradation? So first of all, Dr. Bottini provided us the pegylated single-wall carbon nanotubes. These were 2 kilodalton molecular weight carbon nanotubes. The nanotubes were approximately of 100 nanometer in length, and in height, it was around 1.5 nanometers with the PEG chains on the surface. and they were amino-terminated. So the first thing that we did was an in vivo study in which the nanotubes at the concentration of 5 micrograms were given to mice, 4-month-old mice in the knee joints through intra-articular injection. And after day 1, 3, and 21 days, the mice were sacrificed. The knee joints were fixed and sectioned, and then what we did was to do a label-free confocal Raman spectroscopy-based mapping. The spectra that we selected was the G band, which comes from the tangential CC stretching movement of the carbon on the surface of the carbon nanotubes, and this is a very strong signal. So no biomolecules can actually cross this signal. And as you can see, these are all coming from the confocal microscope. These are light microscope images of the sections. And on the same slides, these are the G-band of the carbon nanotubes that are inside the knee joints. And when we overlay the two images, you can see the nanotubes on day 1 were actually very much localized in the region where they were given to the mice knee joints. But on day 3, the interesting thing that happened was the nanotubes were spread all over the knee joint, the synovial cavity. So this is the synovial membrane, when you can see the nanotubes. This is the meniscus, which also has chondrocytes, along with the synovocytes, and the cartilage region. So the nanotubes were spread, and as previously shown during the study done by Dr. Bottini's group, that within 3 days, the antisense oligomers were delivered. So we found a very similarity between the two studies about the biodistribution. Finally, on day 21, we could see that most of the carbon nanotubes were actually gone from the knee joints, which was very interesting because we could see that the nanotubes have moved out of the synovial cavity into the synovial membrane and actually very deep into the synovial membrane where there are lymphatic vessels present. So we presume that there is a mechanism which is probably coming from the resident macrophages for clearing these carbon nanotubes from the knee joint within 21 days and leading them to the synovial, the lymphatic vessels for clearance from the knee joints. Another thing that we found very interesting was the biodegradation of the carbon nanotubes in these sections. So as you can see, this is the D to G ratio that we normally study in the Raman spectroscopy. The D band comes from the asymmetric mode peak, which represents the oxidative defect sites or the holes that are present on the surface of the carbon nanotube. And this is the tangential CC stretching mode comes from the graphitic structure of the nanotube. So when we take a ratio, we can find the amount of defect sites present on the surface of the nanotube due to oxidative damages. And if we see this graph, you can see that day 1 is basically almost the same, except in the synovial membrane, where there is a slight increase in the oxidative degradation, as we can say, or oxidative damage introduction. But day 3, we see a significant increase in the cartilage region, the meniscus region, which are highly rich in the chondrocytes, and the synovial membrane region, partially, which was very much increased in macrophages. So this proved that there was a degradation happening in the knee joints while the trafficking was also happening together. On day 21, however, we see a decrease in the peaks. This is, if we correlate this to the previous image, this shows that, one, the nanotubes are being cleared out by trafficking, and secondly, possibly also by degradation. So most of the nanotubes that were present were already either degraded or they were trafficked out of the knee joints. To further confirm these studies, we did some in vitro studies where we took the same pegylated single-walled nanotubes and gave them to the ATDC5 cells, which come from murine chondrocytes. And in the first graph, as you can see, after giving the pegylated nanotubes, we saw up to 48 hours an increase in the D to G ratio, intensity ratio, showing that there was a degradation happening in the cells. When the cells were further stimulated with interleukin-1 beta, which is a signaling molecule for production of, for the stimulation of INOS, or inducible nitric oxide synthase, we saw an increase in the defect size at 48 hours. This was showing that when the nitric oxide was probably produced, it was increasing the degradation of the pegylated single-wall carbon nanotubes. Furthermore, at 48 hours, we performed another study where we used the same PNTs and interleukin-1 beta and further added an L-name, which is an INOS inhibitor. And as you can see, that immediately after adding the L-name, at 48 hours, the degradation was significantly reduced, showing that the nitric oxide production in the cells was the responsible mechanism for this degradation. To further isolate out the nitric oxide, what we did was to check for the nitric oxide in the ATDC5 cells. So we stimulated the cells just with interleukin-1 beta with the INOS inhibitor, 2 kilodalton PNTs, or the pegylated nanotubes, and interleukin-1 beta and 2 kilodalton PNTs. And as you can see, the nitric oxide was highly produced in the cells, which was stimulated with the interleukin-1 beta and the interleukin-1 beta and 2 kilodalton nanotubes at 6 and 24 hours, showing that the nitric oxide was the main reason for this whole degradation process. So that was with the chondrocytes. So then we moved on to the macrophages, because that was the cells that we think are responsible for the trafficking. So what we did was to take raw 264.7 cells, these are murine macrophages, and we gave the same amount of nanotubes, that is 10 micrograms to 1 million cells, and saw the uptake first. And as you can see, as compared to controls, we saw carbon nanotubes inside the vacuoles that are probably endosomes. So we can see the carbon nanotubes present inside the cells within the first 24 hours. Also, when we did the Raman spectroscopy, interestingly, as compared to the 48 hours degradation that we found in the ATDC5 cells or the chondrocytes, we did not see any degradation happening in the macrophages. So it was a slow process. However, by 96 hours, we started looking at some biodegradation. So that means that the macrophages were more involved in trafficking, perhaps, as compared to the biodegradation when we compare it to the chondrocytes in the knee joints. To finalize the study, what we did was to take SYN1 or 3-morpholinosidonimine. It's a nitric oxide donor. It's a pharmacological nitric oxide donor. And we simply incubated the nanotubes with this SYN1 in water. And what we found was, by 24 hours, there was an increase in the defect sites on the surface of the nanotubes, which pretty much stayed the same till 48 hours. So whatever damage happened was within the first 24 hours. And if you compare it to the ones that were simply suspended in water, there was no damage that was observed. So coming to the conclusion, what we found was from the in vivo studies using confocal Raman spectroscopy mapping, we saw degradation and trafficking of the pegylated single-wall carbon nanotubes in the knee joints of the mice. In vitro studies further confirmed rapid degeneration of the pegylated single-wall carbon nanotubes by the chondrocytes using nitric oxide as a mechanism and trafficking by the macrophages. Finally, our study paved way for use of functionalized and biodegradable carbon nanotubes as safe nanosystems in the delivery as a treatment in arthritis or osteoarthritis. And finally, functionalized or pegylated carbon nanotubes can be used as a mode for gene and drug delivery in chondrocytes in the disease models. With this, I would like to thank my professor, Bengt Fadil, my colleague, Dr. Pedro Costa, our collaborators from Sanford Burnham Prebys Medical Discovery Institute, Associate Professor Massimo Bottini, Professor Nunzia Bottini, and Dr. Cristiano Sacchetti from Loyola, and from Rice Institute, the collaborators who did the Raman spectroscopy for us, Dr. Birgit de Brandner and Dr. Jens Sommertune, and the funding agencies. Thank you very much.
Kunal, this is fantastic.
Thank you.
So the question is, eventually when you do drug delivery or some siRNA delivery, you usually wrap the carbon nanotube with some DNA fragment or some fragment. Do you think after the wrapping, the whole biodistribution or cell uptake might be changing?
It is possible, and it will also be affected by the biocorona that will be formed after the modifications. What we did here was just to use a pegylated single-wall carbon nanotube for this trafficking study. But definitely when we have some more loaded drug loading or siRNA loading on the nanotubes, it will change the surface, it will do surface modifications, and naturally it will have a little bit more trafficking changes. But we will have to study it probably in the future about that.
Because we published a couple of papers in Nanoletters that if you change a little bit in the wrapping system, even some of the sequence change, trafficking is changing very different. Toxicity changes. I mean, it's a lot of questions. So we are working with Anand Jagota's group as well as Starana's group. We published a couple of papers that simple modification can change the trafficking and as well as toxicity of the cells. So just keep in mind, in future, maybe we can do that kind of alterations of the sequence and see which is best for the therapeutic values.
Sure, we'll take care of that. Thank you very much.
László Dézsi, Semmelweis University, Budapest. Congratulations for the nice presentation.
Thank you.
However, I have a question. For carbon nanotubes, you mentioned in the beginning of your talk that it could activate the immune system. And it's also shown that it could induce the so-called CARPA reaction, the complement activation-related cellular allergy. This is our field. And in addition, there are new data that pegylation, the PEG itself, can do it. So when you combine the two, wouldn't you think it could cause problem, or what did you study on this field?
Of course, activation of the immune cells is definitely necessary, because for the biodegradation, we need activated cells to get stimulated and release these peroxidase enzymes that are responsible. So activation by PEG chains, or by the PEG molecules on the surface of the nanotubes, or nanotubes per se, is required. The thing is that if it is a pristine nanomaterial and it's not biodegradable, then it's a problem, because then it can cause granuloma formation and probably complement activations leading to cancer or fibrosis. So it can happen. But if it is biodegradable within a certain time period, then that doesn't happen, because then the cells, activated cells, will get stimulated, they will release the enzymes, and the nanotube is degraded. So that's the main point of this part.
Okay, but the question is, what is the time frame of this biodegradation? Because this CARPA reaction is a very short reaction. So when you give it intravenously into the circulation, then and there is a sensitive organism, then this reaction starts in minutes. Absolutely.
Well, with neutrophils, what we have found is the biodegradation actually occurs within 8 hours, as we showed with the pegylated nanotubes. And by 24 hours, Kagan et al. Has published with Professor Fadil in 2010 in Nature Nanotech that the complete biodegradation actually occurs within 24 hours. So the nanotubes, if they're oxidized and their surface functionalized, they can be easily removed into other byproducts, and then in carbon dioxide and simple carbon dioxide and water. That's as the last product. Okay. So it can occur within 24 hours, because neutrophils are the most potent cells for these type of reactions. That's what we found.
Thank you very much.
Thank you.
More questions? Well, so the last speaker, Dr. Guo, was not able to attend the conference because of visa problems, so he apologized. Also, his supervisor, Professor Liang, sent me a very nice email. So I would like to conclude here the session about arthritis nanomedicine, and to really warmly thank the speakers that presented the results, also the foundation that made it possible this session, and all of you that were very nice to come here and to listen to our talks. Thank you very much, and see you next year.